Hello friends, as all of you are aware about the polling on the course Antibiotics for Medical Students on various platforms like Facebook, YouTube, Telegram and WhatsApp. So, to all our subscribers who voted yes, your journey starts here. And for those who have not joined the poll yet, don't miss out. Subscribe now for the antibiotics for medical students and take the first step toward mastering the world of antibiotics. So, from today, we are going to start the course, that is, first week lecture will be today on the introduction of antibiotics. We will cover history of antibiotics, discovery and development of antibiotics, classification of antibiotics, and overview of the bacterial infections. Starting from the history of antibiotics, that is a revolutionary journey, antibiotics have had an extraordinary impact on the human health, transforming once deadly infections into the treatable conditions. Their history spans millennia of human experimentation with natural remedies, punctuated by key scientific breakthroughs that reshaped the medicine. This account explores the major milestone in the discovery and development of antibiotics and their role in combating the infectious diseases and the challenges that lie ahead. So, in the history, the pre-antibiotic era, that were the early remedies and intuitions, long before the scientific understanding of infections, ancient civilizations recognized the healing properties of certain substances. These rudimentary practices laid the foundation for what would eventually become antibiotic therapy. Like in the ancient Egypt, the Ebers, an ancient Egyptian medical text, mention the use of moldy bread to treat the infected wounds. This intuitive practice utilized the antibacterial property of the mold, even though the mechanism was unknown. In the China and India, in traditional Chinese medicine, herbal preparation containing plant-based compound with antibacterial properties were widely used. Similarly, in India, Ayurveda, the ancient Indian system of medicine, advocated the use of honey, turmeric, and other substances known to possess antimicrobial activity. In the ancient Greece and Rome, the Hippocrates, who is also known as father of medicine, recommended the use of vinegar for wound cleaning, while Roman physicians applied honey and wine to infected wounds, relying on their natural antimicrobial properties. Though these remedies were effective to some extent, they were inconsistent and poorly understood. The absence of scientific knowledge about microbes meant that these practices relied on observation rather than controlled experimentation. So, from this, after the pre antibiotic era, the concept of germ theory of disease, that was the foundation of modern medicine, that turned the point in understanding infectious disease, came with the germ theory. It was developed in the 19th century. Pioneers such as Louis Pasteur and Robert Koch demonstrated that the microorganisms were responsible for causing disease, providing a scientific basis for targeting the pathogens. Louis Pasteur experiments with fermentation and spoilage showed that microorganisms could be killed by heat, leading to the concept of pasteurization. His work laid the groundwork for understanding how infections could be controlled. The another Robert Koch, he identifies the specific pathogens responsible for disease like tuberculosis and cholera, establishing the idea that each disease was caused by a particular microbe. His postulates become cornerstone of the microbiology. The germ theory not only revolutionized the hygiene and sanitation practice but also paved the way for targeted treatment against microbial infections. After this era, the discovery of antibiotics, that was the discovery of penicillin by Alexander Fleming in 1928. It marked the beginning of modern antibiotic era. This breakthrough was both serendipitous and transformative. The accident, Fleming, a bacteriologist who was a bacteriologist, he was studying staphylococcus bacteria when he noticed that a mold containing one of his contaminating one of his petri dish killed the surrounding bacteria. The mold was identified as Penicillium notatum, and it was active compound of penicillin, which was found to inhibit bacterial growth. In the There were many challenges in the development. While Fleming recognized the potential of penicillin, he lacked the resources and technique to mass produce it. The breakthrough came with a decade later when a team of scientists, including Howard Florey and Ernest Boris Chain and Norman Heatley, successfully purified penicillin and demonstrated its efficacy in treating infection. During the World War II, mass production of penicillin saved countless lives, particularly among the wounded soldiers. It was hailed as a miracle drug and spurred a wave of research into the other antibiotics. From the 1940s to 1960s, the golden age of antibiotics starts. The success of penicillin ignited the flurry of discoveries, often referred as the golden age of antibiotics. During this period, numerous classes of antibiotics were identified and brought into clinical use, like streptomycin, discovered in 1943 by Salman Waxman and his team. Streptomycin was the first effective drug for the treatment of tuberculosis. It also marked the beginning of use of soil microorganisms as source of antibiotics. Then came tetracycline and chloramphenicol. The broad spectrum antibiotics were discovered in the 1940s and 1950s. They could treat a wide range of infection, including respiratory and urinary tract infections. Another drug, erythromycin, discovered in 1952, became an alternative to penicillin for those patients who were allergic to penicillin. It was particularly effective against respiratory and skin infections. The golden age of antibiotic transformed medicine, enabling the effective treatment of infectious diseases like pneumonia, syphilis, and gonorrhea. It also paved the way for complex surgeries and cancer therapies, as infection could be now managed. Then the rise of antibiotic resistance, along with the new antibiotic discoveries, arise. As the use of antibiotic became widespread, so did the emergence of antibiotic resistance. This phenomena occur when bacteria evolve mechanism to survive exposure to antibiotic, rendering treatment ineffective. Early warnings. As early as the 1940s, researchers observed that some bacteria were resistant to penicillin. The widespread and often indiscriminate use of antibiotics accelerated this trend. Resistant strains like methicillin-resistant Staphylococcus aureus (MRSA) and multi-drug resistant tuberculosis (MDR-TB) is become significant public health challenges. These superbugs are difficult to treat and often require expensive, toxic, and less effective alternatives. The misuse of antibiotics and such as overprescription, improper doses, and use in livestock has exacerbated the problem of antibiotic resistance. In some cases, antibiotics are prescribed for viral infections against which they are ineffective. Antibiotic resistance now threatens to undo decades of medical progress. The World Health Organization has identified it as one of the greatest threat to global health. Then the modern advances and future direction for the antibiotics. Despite of the challenges of resistance, research into new antibiotics and alternative treatment continues. Scientists are exploring innovative approach to combat infections. New antibiotics, Teixobactin, offer hope for overcoming resistance. Teixobactin targets bacterial components that are less prone to mutation, making it harder for resistance to develop. Then the bacteriophage therapy use virus that infect and kill bacteria. Bacteriophage can be tailored to target specific pathogens, offering a potential solution to resistant infections. Then artificial intelligence is being used to accelerate the discovery of new antibiotics by analyzing vast data sets to identify promising candidates. Efforts to promote the responsible use of antibiotics are gaining momentum. Education campaigns, stricter regulations, and better diagnostics aim to reduce misuse and preserve the efficacy of existing drugs under the stewardship programs. The future of antibiotics depends on the combination of scientific innovation, global cooperation, and individual responsibilities. The history of antibiotic is treatment to human ingenuity and power to scientific discovery. From ancient healers experimenting with natural remedies to modern researchers tackling resistance, the journey of antibiotic reflects our ongoing battle against the infectious disease. A medical milestone in which the antibiotics have saved millions of lives, transforming healthcare and extending life expectancy, they remain one of the greatest achievements in the history of medicine. A shared responsibility as we face the challenge of resistance, the legacy of antibiotics remind us that the progress comes with responsibility. By using these life-saving drugs widely, we can ensure their benefits for generations to come. Now came to the classification of antibiotics. Antibiotics can be classified based on their mechanism of action, spectrum of activity, and chemical structure. Based on the mechanism of action, these can be divided into cell wall synthesis inhibitors, protein synthesis inhibitors, nucleic acid synthesis inhibitors, metabolic pathway inhibitors, and cell membrane disruptors. Examples under each are under the cell wall synthesis inhibitor, penicillin, cephalosporin, carbapenemase. These antibiotics inhibit the synthesis of peptidoglycan, that is a crucial part of the bacterial cell wall leading to the cell lysis. In the protein synthesis inhibitor, tetracycline, macrolides, aminoglycosides, these antibiotics bind to bacterial ribosome and inhibit the translation of mRNA into proteins. Nucleic acid synthesis inhibitors like quinolones, rifamycins, these antibiotics interfere with DNA replication and RNA transcription. Under the metabolic pathway inhibitors, sulfonamides, trimethoprim, these antibiotics inhibit the bacterial metabolic pathways such as folic acid synthesis. And lastly, cell membrane disruptors include polymyxins, as these antibiotics disrupt the bacterial cell membrane leading to the cell death. We will discuss mechanism of action in the next lecture, that is the second week lecture, in details. And based on the spectrum of activity, antibiotics can be narrow spectrum antibiotics and broad spectrum antibiotics. For general overview, narrow spectrum antibiotics which are effective against a specific bacterial strain. For example, only specific for gram positive or only for gram negative bacteria. Broad spectrum while effective against a wide range of bacterial strains including both, that is gram positive and gram negative. Based on the chemical structure, antibiotics can be classified on their chemical structure, which often determine their mode of action, spectrum of activity, and pharmacological property. So here is a classification by chemical nature. First one is beta-lactam antibiotics. These contain a beta-lactam ring in their structure. For examples, penicillin, cephalosporin, carbapenem, and monobactam fall under this category. Second class is aminoglycosides. These are composed of amino sugars linked with glycosidic bond. These include gentamicin, amikacin, and streptomycin. Third class is tetracyclines. As the name suggests, there are 4 cyclic rings in their structures. Examples of this is tetracycline, doxycycline, and minocycline. Fourth category is macrolides. These contain a large macrocyclic lactone ring in their structure. Examples are erythromycin, clarithromycin, and azithromycin. So, fifth class is fluoroquinolones. These have a fluorinated quinolone structure. Like drugs like ciprofloxacin, levofloxacin, and norfloxacin, etc., fall under this class. Sixth class is sulfonamides containing a sulfonamide group. Example sulfamethoxazole and sulfadiazine. Seventh class is glycopeptides composed of glycosylated cyclic and polycyclic peptides like vancomycin and teicoplanin. Eighth class is oxazolidinones. These are characterized by an oxazolidinone ring in the structure. Examples under these are linezolid, tadizolid. Ninth is polypeptides, which contain a polypeptide chain in the structure. Examples are polymyxin B or colistin, and bacitracin. 10th class is rifamycins. These have naphthahydroquinone structure like rifampin and rifabutin. And the last class under the chemical structure classification is nitroimidazoles. These contain nitroimidazole group like metronidazole and tinidazole. So coming to the our last heading, that is general overview of bacterial infection. So bacterial infection are caused by pathogenic bacteria and can infect various parts of the body. So there are a type of bacterial infection based on the site which they affect. So first one category is respiratory tract infections. Under this the respiratory tract infection can be upper or lower. So examples of respiratory infectious diseases are pneumonia, bronchitis, sinusitis. These are mainly caused by bacteria like Streptococcus pneumoniae, Haemophilus influenza, etc. Next is skin and soft tissue infections like cellulitis, impetigo, abscess. The main causative organisms of these infections like Staphylococcus aureus and Streptococcus pyogenes. Another class is urinary tract infections, that is cystitis, polynephritis. Also, these are caused by the bacteria like Escherichia coli and Klebsiella species. Next example is gastrointestinal infections, that is gastroenteritis, food poisoning. These mainly caused by the Salmonella, Shigella, Campylobacter. Next is sexually transmitted infections, STI. The bacterial infections like gonorrhea, syphilis, chlamydia, caused by bacteria like Neisseria gonorrhoeae, Treponema pallidum, and Chlamydia trachomatis. The diagnosis and treatment of bacterial infection often involve clinical evaluation, laboratory tests, and imaging studies. Treatment typically include the use of appropriate antibiotic based on the type of infection and susceptibility of causative bacteria. So in the today lecture, we discuss from the history, that is the pre-antibiotic era, then the discovery of then the germ theory of disease, then discovery of penicillin, then the golden age of antibiotics, including discovery of streptomycin, tetracycline, chloramphenicol, erythromycin, and the rise of resistance and contributing factors to the resistance, and modern advances and future direction uh, for the antibiotic research. And lastly, the classification of antibiotics, that based on the different types like mechanism of action, spectrum of activity, and chemical structure. And lastly, we covered the general overview of bacterial infections by the system, that is respiratory tract, skin and soft tissue, urinary tract, gastrointestinal infection, STI, cardiovascular, and central nervous system, CNS infections, etc. So in the next video, that is in the next lecture, from in the after 7 days, we will discuss the mechanism of action of each class of drugs in the details. If you have any query, kindly comment or ask in the Telegram group or WhatsApp group. Whether you feel convenient, you can ask your query. If you like the video, then share and subscribe to our channel, and ask your colleagues to also join you on this to this course along with you, and discuss among your friends. Watch the lecture complete. You can't miss any part of this course. This was all about today's lecture, that is introduction to antibiotics is complete today. Thank you.